Thanks. It's a real pleasure to be here today. I'm going to tell you a bit about why we work on aging, and then I'll also tell you, in context, the story of Bioage. And as Tony mentioned, how a really crucial early partnership with the biobank got us some really powerful data for aging. So the reason why I personally work on extending human lifespans is because I think this is the most powerful way to affect human chronic diseases. Now, as you get older, your risk of getting all these different chronic diseases of aging, like cancer, heart disease, Alzheimer's, and diabetes, basically rises exponentially with age. And so does your risk of dying from each one of them. So they're all very clearly age-related diseases. Now, in the United States today, just to warn you, all my figures will be from the United States because that's where we're based, but this is the average lifespan for a woman. It's 81 years of age. Now, what if you could completely cure one of the age-related diseases? So what if we cured all cancer? What would that do to the average woman's lifespan? Well, it would get her to 85. It's not so exciting, right? That's not really that much of a difference. Similarly, if we could cure all of heart disease, that would only add one additional year. Now, here's a different question. For a couple of decades, researchers in labs have been able to make mice live longer, over and over again, using a number of different drugs, a number of different genetic interventions. And what if that lifespan change translated directly to us? What would that do? Well, that would get us to 113, which is crazy, right? And that's an additional 3 decades of healthy life too, not sick life. That's a really important point. And I want to distinguish now between lifespan and health span. We don't just want to live longer, we want to be healthy longer. And right now, again in the US, the average health span is only 62. So from 62 to 81 for 20 years, you're sort of in and out of the hospital with one or more chronic diseases of aging. And we think we can change that by bringing these therapies that impact aging to the clinic. So in particular, in these mouse studies that I'm going to tell you about soon, these animals don't just live longer, they're healthier longer, which is what we all want. Now, despite the importance of aging, it's still largely scientifically unexplored. It's really new territory. And part of the reason for that is that very little money is spent on it in academia and also in biotech. But that's just starting to change right now. So for example, the NIH spends $37 billion annually on scientific research, but only 1% of that goes towards aging, even though it has this transformative potential to impact all these different age-related diseases. And if you look at the Medicare spending as a whole, that's $650 billion. That's a lot of money that goes on mostly late-life care. And if you look at aging research as a fraction of that, of course, it's even more small. It's 0.05%. So I'm going to tell you sort of the history of the field of aging, where we are today, and how Bioage fits into the field. So before the 1990s, aging was kind of a backwater scientifically. No one wanted to work in it. They thought that aging is so complicated, so many different things go wrong with our bodies. How can we possibly expect that we could make a simple change that would dramatically affect lifespan? And that idea suddenly changed in the early 1990s when a couple of papers came out by Cynthia Kenyon, Gary Ruvkun, and Tom Johnson. And they showed that you could take this worm, it's an animal that usually lives 2 weeks, you could delete a single gene in the worm, just one gene, and then the lifespan, then the worm's lifespan would double. So it would live like 4 weeks instead of 2 weeks. And that was really amazing for people. And they thought that no way. And the field sort of flooded with invertebrate geneticists, sort of brought all these genetic tools to study aging in worms, flies, and yeast, and started to map out actually hundreds of individual changes that you could make to these animals that would make them live longer and live healthier. So that's great for flies and worms, but what about mammals? So in the 1960s, a group of Canadian scientists sailed out to Easter Island, and they wanted to sort of study the isolated population there, and while they were there, they collected a bunch of soil samples, and they brought this dirt back. And over the years, people sort of characterized various things in the dirt, and one of the things they found was this compound, this natural compound called rapamycin, which is really bioactive in cells and in animals. And one of the most, basically in 2009, this paper was published showing that if you took middle-aged mice and you fed them rapamycin from middle age until their deaths, they would live 14% longer, which is tremendous. And this is really the first well-replicated lifespan extension in a mammal, in a mouse. It's been done, over and over again with thousands of mice. It's really a strong result. And another reason why I like this example is that it's really, this is the paper, the first example of an aging therapy that's been translated directly to humans, to us, just recently. So Restore Bio is a biotechnology company that spun out of Novartis, big pharma, and just recently they IPO'd in 2018. And also in 2018, so about half a year ago, they published positive phase 2b clinical trial data where they took older people, they gave them this drug, or something very chemically related to it, and showed that people who were on the drug got fewer respiratory infections. So the idea is that it boosts immune aging in people as well. So this therapy was translated all the way from mice to humans in about a decade. Another great example is senescence, cellular senescence. So as you get older, you get more of these sort of senescent cells, which are cells in your body that are sick. There's sort of the gross gray ones here. They're actively secreting these toxic factors. They're making neighboring cells sick. And there was this idea in the aging field that, well, what if we could just kill those cells? What if we just got rid of them? People thought maybe that would make you healthier, maybe that would make you live longer. That was first proven in 2011, and then again in 2015 in mice. So these two mice here are actually the same age, but you can see the guy on the right is the one who has its senescent cells removed and looks way healthier, much sort of sleeker, nicer coat, not losing as much hair, not as fat. And again, so this is the first such paper that was published in 2011. This was translated just recently. So Unity Biotechnology, probably the best known company in the aging space, also just IPO'd, went public in 2018. And in 2018 began their phase 1 study of this second sort of anti-aging drug, which is fighting senescent cells. So you can see this is all just happening like right now. It's all very new and recent. The other finding about aging that we've learned basically from mammals that I'm excited about is blood. And this is related to what we do on Bioage. So there's a lot of evidence that things in blood can impact how you age. And the reason we know this is because of this very kind of Frankenstein experiment, where what you do is you take an old mouse and a young mouse, and you surgically attach them. And that means that they end up sharing a bloodstream. So after about a month, the old mouse is completely exposed to young blood, and the young mouse is completely exposed to old blood. Now, what happens when you do this is that it actually ends up being really, really good for the old mouse, and terrible for the young mouse, as you might expect as well. And specifically, in the old mouse, you see greater blood flow, you see muscle rejuvenation, they smell better, their brains get better, like really across the board. So there's something in the blood that's really good for you. What is it? And this was published. One of those sort of papers that really brought this back was this Nature paper here. So blood works, but what is it in the blood that works, right? So that's partly the idea behind Bioage. We want to find the molecular factors in your blood that can positively impact how you age and turn them into drugs. And how we do that, our approach, we also want to find, I should say, the human blood factors, not the mouse ones, is we look at people who lived, short lives versus long lives. This is just showing you the different distributions of how long people live and see what's different about their blood. And the idea is to start with healthy aging cohorts, look deep in their blood with all the omics technologies that we can get our hands on. We just want to look at every single molecule there is in the blood, so all the metabolites, all the proteins. We also use genetic data and methylation data as well. And from that information, go to new aging targets that we can drug. And as I mentioned, the Estonian Biobank is actually the first crucial partner for us. So I founded the company in 2015 out of my postdoc at Stanford. And a few months later, contacted Tonu, and we built a cohort and found some really interesting signal in it. So yeah, the Biobank is one of the world leaders in this space. It has really great follow-up data for mortality, for aging factors that we care about. And our partnership was actually covered in Wired. It's a really fun story. So this is a sort of an abbreviated version of our our journey so far. So our partnership was in 2016. In mid-2017, a year and a half ago, we raised our Series A, led by Andreessen Horowitz. And based on the data that we analyzed, we patented our first drug in 2018. If everything goes right, we can put that into people in 2 years. Oh, and as of, just yesterday, we announced that we raised our Series B. So in total, we've now raised $37 million to advance these drugs to the clinic. This is sort of how we do this, all packed into one slide. The really key thing for us is to start with healthy aging cohorts that have blood collected a long time ago. We can go in and measure everything in those blood samples and see which of those things predict how long you live. So that's sort of step one. And the right way to do that, of course, is with these really deep omics technologies and with machine learning and AI-based analyses. And we make some predictions, some guesses about which of these factors are actually going to impact aging. And then we test them in the lab because you have to make sure that they're actually real. And how we do that is we take a bunch of old mice. We inject the factor that we think might be good for them, and then we see if they're rejuvenated using a bunch of different kinds of assays that look at frailty and resilience in these animals. Then if we believe the target, we turn it into a drug. So there's really 4 key components to our platform. This one I mentioned before, building the right healthy aging cohorts. We've done this now with a few different partners, like the Estonian Biobank, getting these old blood samples, the older the blood, the better, so you can look at aging throughout the lifespan. So 10 to 45 years is what we have with the cohorts we have right now. We go deep with proteomes and metabolomes, and we layer on lots of additional data like genetics. So these are some of our partners that we use. So the Broad and UC Davis for metabolomics, Sonologic for proteomics. And then, from this, these massive data sets, to find out what the actual relevant potentially causal signals are, and that's a machine learning problem, and that brings you to sort of aging biomarkers. This is just to show you some of our results. It's all unidentified, of course, but just from our proteome and from our metabolome, it's just a plot of effect size. So the things at the top of the graph have the strongest signal for aging. And as you can see in the red dots are things that other pharmas are already working on in the context of age-related diseases. So we recover known targets, but we also find a bunch of new ones, and that's what we're working on right now. From biomarkers, we go to predicted aging targets. And this is just to show you how we use genetic data as well. So we don't just use the proteome and metabolome, we also layer on more information. This is just an example showing our first protein. So not only in our cohorts does it strongly associate with living longer, so people have more of this protein end up living longer. This protein is studded with genetic variants that are associated with your risk of multiple aging-related diseases. So that's one reason why we got really excited about that protein. Because we look at the intersection of all these different data sets, and it was just really strong signal. And then finally, if something looks awesome in silico, you still have to test it out in vivo, and that's why we're a really integrated company with a great data science side, but also our own wet lab, our own mice. And these are our mice in our lab, and some of the assays that we do to evaluate if these are true rejuvenation factors. So really, we're sort of 3 key components at Bioage: data-driven target discovery, in vivo validation, using validated aging assays, like a whole suite of them, and then finally, drug discovery for those targets that we commit to. And I think for the first time, just in the past few years, we're finally in the era of really human aging, not fly aging and worm aging or even mouse aging. And that's really only enabled by the existence of these technologies, which are still fairly new, proteomes, metabolomes, and the right algorithms to analyze them. So just to zoom out again, I'm going to tell you a few of the other exciting discoveries in aging that are coming up. That, things that we're not doing that I also think are promising for aging. So one of those is exceptional longevity. So there are, amongst us, these outliers, these humans that live to be over 100, over 110. And this woman in the middle here is Leela Denmark. And she was a practicing physician, a doctor, until she was 105. So she retired 40 years later than the rest of us do. Human proof that you can live healthier longer. This one here is one of my favorites as well. It's a photo of 4 siblings when they're all under 10, and then when they're all over 100, showing there is a really sort of strong genetic component to heritability. So I think it's going to also be really promising to find these longevity outlier families and find out what's different about them in their genes. Another really promising strategy for finding an aging drug is by mining electronic health records. That's actually how we found metformin. So I'm not sure if you're familiar with the drug metformin. It's a first-line therapy for diabetes. We've been giving it to millions of people over the past few decades. And some computer scientists went back and retrospectively analyzed the electronic health records and found that the diabetics who were taking metformin were getting a lot less cancer than the other diabetics. And we're actually living longer even than the non-diabetics. And based on that evidence, metformin is being investigated as a potentially anti-aging drug, not just a diabetes drug. And there might be other drugs like that out there that we can figure out in the same way by sort of analyzing this data. Finally, I'm just going to end with, if all these things work, what's it going to do to our lifespans? How long are we going to live? Now, I already mentioned there's 3 things that humans are already taking today. 2 of them in clinical trials, the rapalogs like rapamycin, and the senescence targeting drugs. And one that's already, you can get off label today, which is metformin. Now, these are all impacting different aging pathways, and they could themselves already add several years to the average lifespan. And going forward, I think it's an important point that there's no hard biological limit on how long you can live. There are mammalian examples like the bowhead whale, which live well over 200 years on average. And if you look at trees, then you can easily get into the thousands. And that's it. Thank you.
Great. Thank you, Kristen. So,
Thank you.
Question. You're really smart. You got PhD done, in a matter of minutes probably. I'm doing it already 9 years or something. So, and you're also, you just, closed your B round. You're running your startup. You're doing this research everyday basis. It's a very huge topic. It's not just, some mice is running around somewhere. It's like humans, genes, aging. This is what we really care about. Tell me honestly, when you were doing just, science and then turning into an entrepreneur, how did it actually felt to you? Did you find it challenging for you? Do you still struggle? Do you have the struggles?
I've always been, I guess, personally motivated by developing therapies that will actually, go to humans. And I sort of realized over time that academia wasn't the right place for that, that really a company was. And so that's when I made the leap.
All right. So your heart already belonged to entrepreneurship, and through science you found.
I realized that over time. Yes.
That's awesome. So I hope that it really encourages other people who have those ideas in academia, and they think that they can turn their academic life into an entrepreneurial life. So thank you so much for inspiration, Kristen.